This was an amazing biological adaptation that our ancient ancestors had inherited.
In the dense forests of Brazil, a skeleton lay untouched for 9,000 years, its teeth preserving secrets older than the written word. When scientists unlocked its DNA, they didn't just uncover an ancient lineage, they uncovered a riddle in human history. Because this mystery didn't begin in Brazil, it began in the blood of millions, quiet, invisible, and overwhelmingly type O. Invisible marker. In the world of genetics, few puzzles are as quietly persistent and strangely overlooked as the dominance of type O blood among indigenous peoples of the Americas. It isn't just common. In some communities, especially among isolated tribes, it reaches 100%. This level of uniformity is unheard of in large populations. Globally, type O blood makes up about 45% of humans. In North America's Navajo Nation, it exceeds 79%. Among South America's Pima, it approaches complete saturation. At first glance, this might seem like a quirk of inheritance, just one of those harmless curiosities tucked away in medical textbooks. But beneath the surface lies a profound question. How did an entire hemisphere become genetically tilted toward a single blood type? Type O blood lacks both A and B antigens. Medically, this makes it universally compatible for donation. But genetically, it suggests something deeper, something that shaped entire populations across a continent. Scientists have long suspected this pattern isn't random, that it was forged by time, environment, survival, and possibly something even stranger. Because when the pattern becomes this dominant, it's not just evolution, it's history written in blood, one that may trace back farther and in more directions than we ever imagined. Lines written in blood. To understand the mystery, we first have to understand the map of blood itself. Human blood types are defined by the presence or absence of antigens, specific molecules on the surface of red blood cells. Type A carries one kind, type B another, AB carries both, and type O carries neither. This absence gives type O its universal donor status, but it also raises evolutionary questions. Why would a trait defined by lack become so dominant in an entire hemisphere? Across Europe, A and B blood types are relatively balanced. In parts of Asia, type B surges, especially in Central Asia and northern India. But the Americas are different. Here, indigenous populations overwhelmingly show type O dominance. And the further back we look, before colonization, before European mixing, the more absolute that dominance becomes. Among the Inuit of the Arctic Circle, the Maya of Central America, and the Quechua of the Andes, type O blood isn't just common, it's practically the default. This level of genetic uniformity is extremely rare outside of bottlenecked or isolated populations, such as island tribes or long-separated genetic pools. Early researchers believed this might have been the result of a genetic bottleneck, a small founding population carrying predominantly type O alleles, which then spread over generations. But this theory left holes. Why didn't A or B alleles reappear, even in contact zones? And if survival pressures selected for type O, what environmental or biological factors made that trait so indispensable? Blood, after all, does more than transport oxygen. It leaves behind a biological trail, a fossil record of sorts, written in proteins and patterns. And what that trail seems to show is not a neat march from Siberia downward, but something much more fractured, much older, and far less linear. The first footsteps. For decades, the leading explanation for how humans reached the Americas centered on a single path, across the Bering Land Bridge from Siberia during the last ice age. Between 20,000 and 15,000 years ago, glaciers locked much of the world's water into ice, exposing a frozen corridor between Russia and Alaska. According to the Standard Model, small bands of hunter-gatherers followed game across this icy plain, gradually filtering south into what would become the American continents. Genetic evidence supports part of this journey. Mitochondrial DNA from many Native American populations links back to lineages found in Northeast Asia. Linguistic patterns and archaeological sites, like the tools of the Clovis culture, have been used to sketch a rough timeline of movement and adaptation. But the deeper scientists dig, the messier the story becomes. If the Bering Route was the only path, why do some South American genomes show no trace of Siberian ancestry? Why do remains in Brazil date back just as far, or farther, than those found in the north? And how does a near-universal blood type persist across thousands of miles, climates, and cultures without reintroduction of other alleles? The land bridge theory accounts for timing and terrain, but it doesn't account for the astonishing genetic homogeneity across indigenous groups, nor the archaeological finds that seem to predate any migration through the north. It paints a picture of a single road into a vast wilderness, when the real story might be one of multiple migrations, unexpected contacts, and deeply embedded lineages that have long been misunderstood. Because as the evidence begins to surface, it becomes clear. The first footsteps into the Americas may not have come from one direction, or even one kind of traveler. Unraveling through DNA. In the last 2 decades, ancient DNA has transformed anthropology. What once took decades of comparative linguistics or radiocarbon dating can now be reconstructed from the powder of a tooth. Genes have become storytellers, revealing forgotten migrations, hidden intermarriages, and secrets buried in the marrow of ancient bones. Among indigenous peoples of the Americas, geneticists began to notice something unusual. Despite massive linguistic and cultural diversity, the genomes often echoed a surprising uniformity, especially when it came to blood type markers. Across distinct populations from the Arctic to the Amazon, the dominance of type O blood remains startlingly consistent. High-profile cases helped humanize these findings. Ernie Lapointe, great-grandson of Sitting Bull, confirmed his ancestry through mitochondrial DNA, bridging oral history with molecular proof. But beneath these personal connections were larger mysteries. Why did genetic diversity seem so compressed in the Americas, especially compared to Africa or Asia? And why did some South American genomes contain sequences that defied the Siberian migration model? The technology allowed scientists to detect subtle patterns, fragments of ancestry that didn't match known lineages from Asia or Europe. These were not recent anomalies. They were ancient signals, pointing to population histories that had been erased, overwritten, or simply never imagined. The genome was beginning to whisper what the old migration models had missed, that the peopling of the Americas might not have been a single chapter, but an entire epic, filled with forgotten routes, collisions, and genetic ghosts waiting to be named. The twist beneath the jungle. In 2020, a team of researchers published a study in Cell that sent ripples through the scientific community. It centered on a cluster of 9,000-year-old skeletons unearthed in Lagoa Sana, a cave site in eastern Brazil. These ancient individuals were among the earliest humans found in South America. But it wasn't their tools or burial positions that stunned researchers. It was their DNA. When scientists sequenced the genomes, they found something no one expected. A genetic signal linking them not to Siberian or East Asian populations, but to indigenous Australians and Papuans. This was not contamination or coincidence. It was a real, measurable ancestry component, one that had persisted for thousands of years and was now coded into the bones of Brazil's oldest inhabitants. The implications were staggering. If these individuals bore genetic ties to peoples of Oceania, then the history of migration into the Americas wasn't just over land. It may have included oceanic routes, coastal drifts, or earlier crossings entirely erased by time. It also suggested multiple waves of migration, not all of them from the north. The neat land bridge-only model was no longer sufficient. And here's where the twist connects. Such a deep, separate lineage could help explain the mysterious dominance of type O blood across indigenous populations. If early migrants carried this trait, and if their descendants became isolated or widespread before other lineages arrived, it would have set the genetic foundation for what followed. An invisible legacy stretching from the South Pacific to the Andes, hidden in plain sight for millennia. This single finding, buried in a Brazilian cave, reframed everything. It wasn't just a matter of where the first Americans came from. It was a question of how many stories had been lost in our assumption of just one, the legacy inside us. Today, the legacy of type O blood is more than a genetic curiosity. It's a living inheritance, quietly shaping lives across the Americas. In medical emergencies, it's often the blood that saves lives first. O negative, in particular, is the universal donor, compatible with all other types. In indigenous communities, this prevalence has been both a blessing and a challenge. the most needed blood type is often the most difficult to collect and store in regions with limited infrastructure. But beyond its role in transfusion medicine, type O blood may offer clues to ancient adaptations. In high-altitude regions like the Andes, its thinner viscosity may have enhanced oxygen delivery in low-oxygen environments. In isolated communities, it may have helped reduce exposure to certain pathogens that targeted A or B antigens. There's even evidence that cultural cooperation, where individuals with universal donor blood were more valuable in childbirth and injury, may have conferred social advantages. And yet, what makes this legacy extraordinary isn't just its survival advantage. It's the possibility that it carries a story older than the continents we stand on. A story of people who crossed oceans before maps existed, whose genetic signatures now hum quietly beneath the skin of their descendants. When we see blood type as more than a category, it becomes a map, one written in migration, adaptation, and invisible threads of ancestry that connect us across oceans and millennia. Because sometimes, the most ancient stories are the ones we carry without knowing. A single drop of blood carries more than oxygen. It carries memory. Type O tells a story few ever hear, yet all of us might share. If this journey moved you, subscribe for more untold histories, and share this with someone who still thinks blood is just biology.